and see how the progression of it differs and stuff.
All right, so good afternoon, everyone. Okay, here. So we're gonna continue on from where we left off yesterday. Don't forget, if you're submitting a rebuttal to the term quiz 2, that rebuttal is due by the end of the day today. So just keep that in mind. And that's all I really have to say. So are there any questions about things before we start? I mean, there never seem to be questions. I don't know why I keep asking, but one day there'll be a question. So, right? Always, this Friday group is the best. They're always the most cooperative. Okay. All right. Okay, so we'll roll on here. We were talking about resting membrane potential in the lecture yesterday, and we got to this slide. And so what we said when we got to this slide is we said, okay, we've got these three major ions that are contributing to the resting membrane potential. We noted how each of them has a different equilibrium potential that it's trying to get to. So sodium is trying to get to positive 72, and potassium is trying to get to negative 89, and chloride is trying to get to negative 84. But there can only be one overall membrane potential. So if all these ions are trying to move across the membrane to bring the membrane potential to their own equilibrium potential, how does this get resolved? And a good analogy, I think, for this, and this was published in, I think, like American Biology Teacher journal at one point. I kind of like this analogy, is they likened it to what happens if you've got a male and a female living together in a home. And as you know, if you've ever lived with people of the opposite sex, females always want the room temperature to be higher because they're always cold. And males always want the room temperature to be lower because they're always hot. So here they're saying we've got this female who wants the room temperature to be 85 Fahrenheit. Remember, American journal here. And you've got this male who wants the temperature to be 65 degrees Fahrenheit. So how do they figure out what the actual room temperature of the house is going to be that they share? And they say, well, let's set the temperature based on the proportion of the rent that we each pay. So if we each pay 50% of the rent, then let's set the temperature to be exactly 50% between our two desired values. So again, neither of us is like perfectly happy because neither of us is getting the exact temperature that we want. But this seems like a fair way. We're kind of setting the temperature right in between what we both want because we're each paying 50% of the rent. By contrast, let's imagine that the male is actually making more money, so he pays more of the rent. And the female is paying less of the rent. And so in that case, well, the male's going to get to have the room temperature be closer to what he wants. He pays more of the rent. He should get the room temperature to be closer to what he wants it to be. So in this case, they've set it to 70 degrees Fahrenheit. And of course, if it was the other way around, if the female was making more money and she was paying more rent, then we would shift the we would shift the temperature in her favor. So the basic idea there is whoever is paying more of the rent gets more of a say in terms of what the overall temperature is going to be. And we basically apply that same philosophy here to these ions. Now, ions are obviously not paying rent, so what is the kind of analogy here? Well, the analogy is how many leak channels do they have, which is kind of represented here by the width of these arrows. The more leak channels you have, the more of a say you're going to get, because you can move across the membrane more easily, the more of a say you're going to get in what the value of the membrane potential is going to be. So given what we talked about yesterday, that there are about 100 potassium leak channels on average, and 10 chloride leak channels on average for every 1 sodium leak channel, we're basically saying that potassium should get the biggest say in terms of what the resting potential is going to be. Chloride gets the next biggest say, and sodium gets the least say because it has the least number of leak channels. And so let's kind of see how this plays out and how we kind of work this out. So I want to introduce you to another equation at this point. We learned about the Nernst equation yesterday, which allowed us to calculate the equilibrium potential for a single ion at a time. But now we're going to get introduced to the Goldman equation. This is going to allow us to calculate the resting membrane potential that results when all of these different ions are kind of simultaneously moving across membranes trying to get to their membrane potential or their equilibrium potential. And so what you'll notice here, if you look at the Goldman equation, is you've got your membrane potential value on the left. We've got some similar or familiar looking terms on the right, gas constant, Faraday constant, temperature, all the same things that we saw in the Nernst equation. But if we look in the parentheses now, what you'll notice is for each of these 3 major ions, we have their concentration outside and inside, okay? And that's because that dictates what their equilibrium potential is going to be, as we talked about yesterday, right? Your equilibrium potential is determined by your concentration gradient. Just note here that the way the Goldman equation handles negatively charged ions is you flip the inside and outside concentrations. So you'll notice that for potassium and sodium, the outside concentration is in the numerator, but then it flips for chloride. That's how this equation handles the negative charge of chloride. But then you'll also notice that each of these concentration values is multiplied by the ion permeability, right? It's multiplied by how permeable is this ion, how many leak channels are there for this ion, because as we just said, that dictates how much of a say this ion gets in terms of determining what the overall resting potential is. So if we, again, do the calculations here, again, I've just put in the same gas constant and Faraday constant that we had yesterday in the Nernst equation, same 310 Kelvin temperature, human body temperature. But here, I put in the same concentrations for potassium, for sodium, and for chloride that I did yesterday. But then what I've put in for these permeability values is just their relative permeabilities, right? So I put a value in for 1 for sodium. We said there are 10 times more chloride channels, so a value of 10 for it. And there are 100-fold more potassium channels, so I put in the 100 there for that. And when we do the calculation now, we get the membrane potential of this cell, which is negative 82 millivolts. So we can see that that value is certainly much closer to the potassium and chloride value than it is to the sodium value, right? And that makes sense because potassium and chloride, way more leak channels for those 2 ions. They get to have way more of a say in terms of what this membrane potential is going to be. So that's the first thing we can appreciate. But the next thing we can appreciate here is that negative 82 millivolts, none of the ions has an equilibrium potential of negative 82 millivolts. So what we have to appreciate here, as it says, is that under these conditions, right, under these conditions that we've just simulated here, none of these ions, neither sodium nor potassium nor chloride, none of them are currently happy with the situation. None of them have their equilibrium potential being met, right? Chloride's pretty close, potassium is kind of close, sodium for sure is unhappy, right? Sodium is way off its equilibrium potential. And so what we have to appreciate is the resting membrane potential is a non-equilibrium steady state, right? It's not an equilibrium state, right? None of these ions is at its equilibrium potential, okay? So it's a non-equilibrium state, but we also call it a steady state because this negative 82 will hold pretty constant at that value as long as these ion concentrations and temperature prevail. But so it's a steady state, but it's not an equilibrium state. And of course that makes sense when we think about our conversations from a couple of days ago. We noted that the resting membrane potential kind of also represents a source of energy to the cell, right? That alone could tell you it's not an equilibrium state because by definition, a state at equilibrium cannot yield energy, right? So if resting potential were an equilibrium state, there would be no way for the cell to use it to actually derive energy from it and power any activity in the cell. So we describe the resting potential as a non-equilibrium steady state. We have to recognize, therefore, that all these ions are trying to move across the membrane in one direction or the other, trying to get to their particular equilibrium potential. But of course, every movement of one ion is going to be countered by a movement of another. And that's what kind of keeps us in the steady state, right? It'd be almost as if, you guys were leaving the classroom, right? But then the first years are following you in behind. So even though, there's movement, you guys are all leaving, but they're all coming in. If we just looked at the number of students in the classroom over time, it would be constant. One person goes out, one person comes in, one chloride moves, a sodium moves in the opposite way, and ultimately everything stays kind of in this steady state value. Now the other thing that is important for us to also note here, connecting back to what we talked about yesterday, is the Goldman equation, which is, again, incredibly commonly used for calculating membrane potentials. As it says here, the equation doesn't count for the electrogenic pumps and any contribution they might make to the resting membrane potential. Now, again, we noted yesterday that in the context of especially neurons, the electrogenic pumps make such a negligible contribution anyways that it's okay to ignore them. But it's worth pointing out that let's say you were to do the math, you were to calculate the resting potential using the Goldman equation, using your known values of temperatures and permeabilities and ion concentrations, and you calculated a resting membrane potential that was way different from the actual measured value. One of the first thoughts that should go through your head, assuming you haven't fucked anything up in terms of like doing the math or making your measurements, the first thing that should go through your mind here is, oh, maybe in this circumstance, there is a contribution being made by these electrogenic pumps, right? Because the equation doesn't account for that contribution, but in reality, there could be a contribution. So that'd be the first thing to think about. If you see, a calculated value of the resting potential not matching with the actual measured value, think about the possibility that your sodium-potassium ATPase or your calcium ATPases might, in the circumstance you're investigating, actually be making a significant enough contribution to account for the discrepancy. The last point I think I want to mention here with respect to resting membrane potentials is the fact that we have these ions that are moving across the membrane, and we said that it's the movement of these ions across the membrane that ultimately is causing the resting potential to form. How do we make sure that the concentrations of these ions don't then change over time? If you've got sodium, for example, trying to constantly come into the cell here, trying constantly to get the equilibrium potential to that positive 72 value, how do we make sure that as sodium comes in, it doesn't eventually cause the extracellular sodium level to drop and the intracellular sodium level to rise, and then that would change the sodium concentration gradients here, and then change its equilibrium potential. And ultimately, if we just let all these ions kind of drift across and reach equilibrium of their concentration, there'd be no equilibrium, or there'd be no resting potential anymore at that point. There'd be no gradient to drive the move of these ions. What helps us keep these ion concentrations constant over time Well, it's the action of, the sodium, potassium ATPase, and I suppose the calcium ATPase too, if we're thinking about calcium. But it's important to recognize that this sodium potassium ATPase, is constantly operating in our cells, right? Because we constantly have these sodiums and potassiums, for example, diffusing across the membrane down their concentration gradient, creating these electrical gradients that we call the resting potential. And we have to be constantly pumping those ions back across the membrane in order to maintain constant concentrations of these ions, in order to maintain that same level of ion flow to allow us to maintain this constant value for the resting membrane potential. And we, again, sort of saw that yesterday anyways, right? We saw that if you inhibited the sodium-potassium ATPase with wobane, that you would eventually get this slow drift of the resting potential to a 0 value. We don't want that to happen, right? We want to be able to maintain a constant resting membrane potential value over time. So we're going to move on now from resting membrane potential. We're going to move on to graded potentials. And so I have a definition here. So it says graded potentials are local changes in membrane potential that occur in varying degrees of magnitude in response to a stimulus. All right? Local, so they really only, we'll see this in a minute here, but they can really only travel short distances before they kind of die out. We're going to see later that's very much in contrast to action potentials, which we can kind of conduct for meters at a time. They occur in varying degrees of magnitude. So I kind of hinted at this the other day, and we'll see it again here later, that this is the reason we call them graded potentials, right? Because there are gradations in their value, and they parallel the gradations in the stimulus that's causing them. So if it's a light stimulus that's causing this graded potential, a dim light might cause a small graded potential, but a bright light would cause a much bigger graded potential. And then, of course, they're occurring in response to some kind of stimulus, right? And again, we defined the stimulus the other day, a detectable change in the internal or external environment, right? And again, we link back to the idea that there are receptors that must exist within the neurons to detect these stimuli and then transduce them into this electrical phenomenon, the graded potential. If we go back to the Goldman equation that I just introduced, right, one of the things we can kind of extrapolate from the Goldman equation is that if the membrane ion permeability changes, so too does the membrane potential. Right, if I go back real quick to our Goldman equation here, we can very quickly recognize that if we change the permeability for any of these ions, right, if we have new channels that open up that allow this ion to pass through the membrane to a greater extent, that's going to lead to the math here mathing differently, right? It's going to lead to a change in the value of the membrane potential. So we can appreciate here that if we change an ion's permeability in the membrane, the Goldman equation predicts that that should lead to a change in the membrane potential. So we're going to talk more about that too. And then last thing here, just in terms of kind of definitions, is the definition of an excitable cell. So it says here, excitable cells can change their membrane potential by changing their membrane ion permeability. So we discussed last week, or earlier this week, that all cells have a resting membrane potential. That's not a unique property of neurons. All cells have a resting membrane potential. However, only a subset of our cells are excitable. Only some of our cells, neurons being a great example, but also think skeletal muscle cells, for example, only some of our cells can change the value of their membrane potential by changing how permeable they are to these various ions like sodium, potassium, and chloride. Okay? So that's what makes you an excitable cell, your ability to change your membrane potential by changing your membrane ion permeability. All right? So just a few definitions to kind of get it out of the way there. So, excuse me, so let's talk now about graded potentials and how does a stimulus ultimately lead to a graded potential. Well, the answer is that we create graded potentials through what are known as gated channels. So again, yesterday we got introduced to leak channels. Leak channels, we said, by definition, are always open. So they're always allowing for ions to move across the membrane. Gated channels, as you can probably guess from their name, they have a gate. And of course, the key property of a gate is it can be opened or it can be closed. And so gated channels, when the gate is closed on these channels, they don't allow the ion to flow through. But if you open the gate, well, then the ion flows through. So right away, we can see how we can change the membrane ion permeability using gated channels by controlling whether the gate on these channels is open or closed, right? So I have here this just figure showing us a few examples of gated ion channels that we find in our nervous system. I'll start over here on the right. So here they're talking about mechanosensitive ion channels. So these are ion channels that are closed when there's no mechanical stimulus present, right? And then they open in response to a mechanical stimulus. And what do we mean by a mechanical stimulus? We mean things like touch, pressure, vibrations, right? You've probably got some mechanosensitive channels that open when you're in this lecture hall, when you hear what seems like jetliners taking off from the top of the building all day long. I don't know what the sounds come from, or whether I'm the only one hearing them. Are you hearing these sounds too? Okay, good. Because there are times when I wonder if I'm just getting old and like hearing things and losing my mind slowly, but good, okay, you hear it too. So those are mechanical stimuli, touch, pressure, vibration, those sort of things. So again, these channels are closed in the absence of those stimuli, but they open in response to those stimuli. And then depending on the particular ion that this channel allows to move through it, we're going to see that as we have this mechanical stimulus occur and it opens this ion channel, and now this ion starts going through the membrane more easily, well, that should lead to a change in the membrane potential, right? It should actually cause the membrane potential to move closer to the equilibrium potential of whatever ion is passing through here. Because now that ion is getting a greater say in terms of what the membrane potential is. In the middle here, they have what they call a ligand-gated ion channel. So a ligand is just like a small chemical that binds to something bigger. So, I mean, a great example of a ligand-gated channel would be a neurotransmitter-gated channel, right? Where you have a neurotransmitter binding to your channel. And again, in the absence of the neurotransmitter, the channel is closed. In the presence of the neurotransmitter, the channel opens. And then once it opens, it allows for some ion to pass through. And now that ion is going to move the membrane potential closer to its own equilibrium potential. Then, of course, you've got voltage-gated ion channels, which we're going to get way more familiar with next week. These open in response to a change in membrane potential. So there's kind of a circularity here of sorts. But you can see here they're showing this channel. It's up here, sorry. Positive on the outside, negative on the inside, so our typical resting condition. But they show here that if you reverse that, if something happens to cause it to become negative on the outside and positive on the inside, that's the stimulus that causes that channel to open, right? So presumably some other thing had to cause that reversal of membrane potential, and then this channel opened up in response and starts to allow some ion to pass through. Again, these are just a handful of channels. I mean, I put it here at the top. There are temperature-gated channels, for example, and basically for any stimulus you can imagine that we can respond to or do respond to, there's a corresponding channel present in our neurons to allow us then to, again, transduce that stimulus into a graded potential, right? And that's, again, the thing I want to emphasize here, right? Neurons speak in terms of electrical conduction. So we want to translate a stimulus into an electrical signal. The graded potential is that electrical signal. Okay. Okay, so just to go through this again kind of very kind of explicitly and clearly, we're going to think here for a moment about what happens if we have a gated sodium channel, right? So a sodium channel that's gated and opens in response to some stimulus. What's going to happen to the membrane potential if we trigger the opening of a gated sodium channel? All right, well, we know that as soon as we open that channel, we're going to increase the membrane permeability to sodium, right? That's what's going to happen, obviously, if we open that channel up. So here we see this figure starts us at the resting membrane potential, right? Typically around -70 in a neuron. And then they show that at time 1 millisecond, you expose this cell to a stimulus, and this stimulus caused the opening of gated sodium channels. And so, again, as I said, that increased the membrane permeability to sodium. And so what's going to happen to the membrane potential in response? Well, now because sodium can move through the membrane more easily, it's going to try and push the membrane potential closer to its desirable value, its equilibrium potential. Its equilibrium potential, as we calculated the other day, is a very high positive value. So look at how the membrane potential responds. It moves in a positive direction, right? Sodium has more of a say now because the membrane permeability for sodium has increased, and so the membrane potential moves in the direction towards the sodium equilibrium potential, which in this case is in the positive direction. And so we get a change in membrane potential, a graded potential, that we specifically in this case call a depolarization. Right? Remember that when you have charge separation across the membrane, that is called, polarized, because you've got kind of a positive side and a negative side. You have these two poles. Here, because you're pushing closer to a zero value, right, we call it depolarization. It's almost like you're moving towards that zero value where there would be no polarity. It's a depolarization. All right? Now, we could also think about this in mathematical terms if we wanted to. Here's our friend the Goldman equation again. And so in this case, and we just did the Goldman equation a minute or so ago, everything here is the same as it was when we last calculated it, except for the fact that we've increased the sodium permeability. I've just taken it from a value of 1, and I've just made it 10-fold higher. Very arbitrary, but a 10-fold increase in sodium permeability because of this stimulus that we subjected this neuron to. And if we redo the math, you now see that the membrane potential is -54, not the -82 that we had calculated earlier. So indeed, whether you want to think about this kind of mathematically or just more conceptually, we can see here the effect of opening a gated sodium channel is the membrane potential shifts in the positive direction. It becomes more positive as a result, okay? We'll do the same thing for gated potassium channels. So let's imagine that we have a stimulus that's going to cause the opening of a gated potassium channel. That is going to cause the potassium permeability to increase, obviously. And so what's going to happen? Well, then potassium is going to have more of a say now in terms of what the membrane potential is. Potassium wants the membrane potential to be down there at -89, as we have seen. And so when we open up these these gated potassium channels, we see the membrane potential shift now in the negative direction, right? It's shifting towards potassium's equilibrium potential. And so this we refer to as a hyperpolarization because we're moving the membrane potential away from zero, further away from zero. So we're making things more polarized. We're hyperpolarizing this membrane. And again, we can look at this mathematically. We can get out our Goldman equation, and we can change the permeability of potassium so it's 10-fold higher, right? Changed it from 100 to 1,000. And if we do the math, we'll get now a resting potential of -88, right? Which is, again, more negative than the -82 that we calculated before. So yeah, the idea here is that we've opened up this gated potassium channel and the membrane potential has moved towards the equilibrium potential for potassium, right? Because we're giving potassium a greater say now in what that membrane potential is because we have increased its permeability. One more time, just to really drive this home, let's look at what happens if you open gated chloride channels, right? Well, obviously we increase the permeability for chloride, so chloride is going to get more of a say now, and chloride has a equilibrium potential of -84 as we had calculated before. So again, we're going to shift from our resting potential towards the equilibrium potential for chloride. So in this case, because chloride's equilibrium potential was more negative, the membrane potential shifts to be more negative. So this will be another example of a hyperpolarization. And again, if we were to do this mathematically, you see what we get here. Now, one point I will drive home here is that the math that we did here with chloride is probably a better representation than this figure. And what I mean by that is, if you think about what happened to our resting potential here, you know, our potential was -82 when we calculated the resting potential. When we increased the chloride permeability by a tenfold value, as we did here, it only shifted it by a millivolt. It shifted it to -83, not really much of a change. And that's actually quite common. It's quite common for chloride to not really change the membrane potential when you increase its permeability. When you open these gated chloride channels, they don't really shift the membrane potential too much. But what they tend to do is stabilize the membrane potential at that resting value, right? Because they make it harder for other ions to kind of push it away from that resting value. So, yeah, that's generally what chloride is going to do, right? It could hyperpolarize, but it very often just stabilizes that resting potential. Now, of course, I want to throw a caveat out here, which is all of these things I just said here, I mean, these are the typical cases, right? And obviously, if you had very different resting potentials and very different equilibrium potentials because you're under some very different set of circumstances, the major overall point to keep in mind here is that whatever the equilibrium potential is for an ion, when you open that ion's gated channels, the membrane potential is going to shift in the direction of that ion's equilibrium potential. Right? So if for some reason, you know, we had different circumstances with totally different concentrations of these ions, right? You have to be mentally prepared to say, hey, I can't just blindly follow what he said here, you know, these specific things. I have to, you know, kind of use the more general principle here that, okay, this is the resting potential, this is the ion whose gated channel is opened, this is the equilibrium potential for that ion right now. The membrane potential is going to shift towards that equilibrium potential value. Okay, question. Yeah, so why, she asked the question like why, for example, here, you know, we increased the chloride permeability, why didn't it actually drop down to hit that red line, and then also I guess why it came back. It came back, that's the easier question, because they actually stopped the stimulus at the, like, wherever it stops being colored here. Like, they only held the stimulus in place for 2 milliseconds. So as soon as you stop that stimulus, then that potassium channel closes, and so the potassium, or sorry, in this case the chloride, you stop that stimulus, the chloride channel would close again, so now you're back to the resting chloride permeability, so the membrane potential should return to its resting value. But why didn't it touch that red line? The answer there is because you still have these other ions that are trying to push things towards their equilibrium potential. So even though you've made it easier for the chloride to get closer to its equilibrium potential, it doesn't get all the way there because you still have those other ions that are trying to push towards their own equilibrium potential. Okay? And that's especially the case if we go back. I mean, that was especially the case with sodium, right? Like, I mean, it didn't get anywhere close to its equilibrium potential, but that's because even though we, you know, changed its permeability, you know, from 1 to 10, I mean, it's still, you know, potassium is still the dominant determinant of the membrane potential here, so we're still going to see the membrane potential be much closer to the potassium value. Yeah, no problem. Okay, where were we here? Here, okay. All right, so I want to talk next about some properties of gradient potentials that are noteworthy. The first property I'm going to talk about here is, and I kind of mentioned this before, is how the magnitude, but also the duration of a gradient potential is directly proportional to the strength and the duration of the stimulus that's causing it. The figure that I have kind of talks about this in the context of muscle spindles. So this seemed like a good opportunity just to kind of briefly talk about muscle spindles and kind of tuck a little extra information into the lecture here. Muscle spindles are these sensory structures embedded within your muscles that basically measure how stretched your muscles are, right? And this is important because obviously, muscles, if you stretch them too much, I mean, they could eventually experience a tear, right? I mean, they have elastic components in them, so those elastic components could tear, and even just, I mean, the other muscle proteins could get, extended to the point where they could tear. So we need some way to determine how stretched out our muscles are, just so that if they are too stretched out, we want to induce the contraction of that muscle to kind of, shorten its length back and prevent it from overstretching and becoming damaged as a result. So that's what the muscle spindles do. The muscle spindles are comprised in part of muscle fibers. So this picture actually tells us that there are two kinds of muscle fibers that we find in skeletal muscles. The ones here that they call extrafusal fibers, like these are the muscle fibers that you think about when you think about muscle fibers. Like they're the ones that actually, do contraction to generate force so you can, pick things up and push things and whatever. So extrafusal muscle fibers are just your typical contracting, force-generating muscle fibers. But then you've got what are called these intrafusal fibers, right? And you can see that they actually make up the muscle spindle, and they're connected to these sensory nerve fibers. So the muscle spindle is actually partly derived from these like specialized muscle fibers called intrafusal fibers. And the basic idea is that when these intrafusal fibers get stretched out, as the muscle is getting stretched out, the more stretched out they are, the more they'll start to release neurotransmitters, and then those will stimulate the sensory fibers, and then those sensory fibers will relay that stretch information back to your brain. And ultimately then you're able to, your brain is able to keep track of how stretched out your muscles are. Again, this is necessary, not even just for the protective mechanism that I was talking about, but even just, for your brain to know, like, hey, is my arm, tucked up against my body right now, or is my arm stretched out, right? It can use the stretch of the muscles here just to figure out your position of your limbs. And we'll talk more about this later, but that's important too, right? Because if I want to, point to the screen, I need to know, like, where's my arm right now? Is it here? Because then I have to do this kind of a motion. Or is it down here and I have to do this kind of a motion? We need to know how our limbs are positioned in space in order to make good movements in terms of the kind of movements we want to make. So in any case, you've got your intrafusal fibers there. The more they stretch, the more they send out neurotransmitters to stimulate those sensory fibers. Those sensory fibers then get stimulated and excited by those neurotransmitters, and they relay that information back to the brain. One kind of interesting thing about these intrafusal fibers is they still do have contractile capacity, and so you can actually use that contractile capacity to adjust their baseline tension. What I mean by that is, let's say you want your muscles to be, only able to be stretched out a little bit before they're going to kind of contract in response. You can have those intrafusal muscles and kind of, actually maybe we'll go the other way. Let's say you want to allow your muscles to get way more stretched out than normal. You can have those intrafusal fibers contract, and then they'll kind of, squeeze that whole muscle fiber, that whole muscle spindle a little bit smaller, and then it will take more stretching of the muscle to actually generate the same kind of response, right? So if you imagine there that whole kind of light pink muscle spindle in the center there, if we had those intrafusal fibers contracting, it'd be squeezed down to this shorter length, right? And then the muscle could actually stretch a lot more before it would really send out a major kind of neurotransmitter signal to simulate or to stimulate those fibers and tell you that the muscle is too stretched. So you can relax versus contract those intrafusal fibers to adjust the kind of baseline sensitivity of that muscle spindle. In any case, I want to show you the data on the right here, because this is the point that the part that illustrates the point that I really want to make. So on the left here, you can see they're showing us the amplitude and duration of this stretch stimulus on these muscle spindles. And I'll just mention, this is the common way that these kind of stimulus diagrams are shown. So when the line is kind of down here low, it means there's no stimulus. And then when you start the stimulus, it kind of shoots up to this higher position and then comes back down when the stimulus is over again. You'll see these kind of diagrams for this. So here we can see that right at this time, that's when someone stretched this muscle. So it stretched it a little bit, right? They stretched it a little bit for a short period of time, and then they relaxed it. Well, notice that the corresponding change in membrane potential exactly reflects the amplitude and duration there, right? That you had a little bit of an increase in the membrane potential for a short period of time, and then it returned back to rest. If you look here at the middle one, here they're going to stretch the muscle twice as much for the same duration, and then let it go. And notice that you get a much bigger change in the membrane potential here for the same length of time, and then it returns back to rest. And then in the third case, they're going to stretch that muscle quite a bit, but for a long duration of time, and then let it go. And correspondingly, we see here the gradient potential is big, and it persists for twice as long now before it comes back to rest. So gradient potentials will always mimic, in terms of their magnitude and their duration, they will always mimic the magnitude and the duration of the stimulus that is causing them. One other small detail that you may have noticed here is that you will notice this kind of drift, right? That over time it seems like the magnitude of the gradient potential is kind of getting a little bit lesser and lesser, even though the stimulus strength isn't changing. This is kind of a habituation response, right? It's a way for the body to sometimes be able to, kind of habituate to a piece of sensory information over time. An example of this would be that, if someone were to, again, maybe you came into the lecture today and you sat down next to someone who had terrible body odor, right? And you're like, oh God, I smell that right away. But then maybe, 5-10 minutes later, you're like, oh, I don't notice the smell as much, right? That's kind of an example of kind of a sensory habituation. Now that can happen kind of in the brain. There are brain regions that can filter out that sensory information, but sometimes it does happen at the level of the actual sensory receptors too, right? That they become less responsive over time to that stimulus, and then that gradient potential in response to the stimulus kind of fades. And you imagine that if we kept this stimulus going for long enough, eventually the gradient potential would just drift back into its resting value, and you'd no longer have a gradient potential to stimulate any action potentials, and you'd no longer be responsive to that stimulus, even though it might still be there, right? And so, that's a good thing to some extent, right? We don't want to be reminded of the smelly pier the entire hour that we're here. Our nervous system in some ways has evolved to say, hey, if you're not responding to a stimulus, then it probably isn't so vitally important to your survival that you need to be consciously aware of it the whole time, right? So if the body odor, you're made aware of it when you first sit down, but if you sit there for the entire hour and you didn't die as a result of smelling the body odor, your brain is like, hey, your nervous system's like, hey, we don't need to constantly make you aware of this. We can allow for this habituation response to stop allowing you to perceive that stimulus. Okay, another characteristic of gradient potentials that we need to be aware of is that gradient potentials, they undergo this phenomenon called decremental conduction. There's kind of two parts to this. So one is the conduction part, which is to say that gradient potentials have the ability to travel from where they originate to other parts of the dendritic zone. So that's the conduction part. The decremental part refers to how the magnitude of a gradient potential actually decays as it is traveling. Okay, so I'm going to kind of try to walk you through how this all happens. So here we're looking at, this can be thought of as a dendrite here. And so we've got in this location some kind of gated sodium channel. And so in response to a stimulus that causes that gated sodium channel to open, you can see that now there are positively charged sodiums crossing through that open channel, coming into the intracellular fluids of that dendrite. And obviously that's going to start to cause the membrane potential in that region to become more positive, okay? Now, just a bit of terminology here. When you have a charged species, like an ion, flowing from one place to another, the term we use to describe that is current. Okay, I throw that out there because it's a term that gets used a lot in nervous physiology. Current refers to the flow of a charged species, like an ion, from one place to another. So here we've got the sodium ions flowing through this open gated sodium channel. And as it mentions here, they are depolarizing this region right where that channel is located. But here's the thing. As those positively charged sodiums come into the intracellular fluid in this dendrite, if you kind of put yourself in their shoes for a second, they look to their left and they look to their right, and they see something they like. They see negative charges, right? So they are attracted to those negative charges. It's kind of like if you imagine, like, you know, like a teenage boy walks into a room, you know, and he's like, oh, there's like pretty girls over here and pretty girls over here. He's not just going to stand here. I mean, he probably will because he's a teenage boy. But let's assume that he's got an actual, like, I don't know, charm and riz. Is riz the word I want to use here? He's got riz. So he's like, hey, I'm going to actually go and move to where these attractive, you know, girls are. The sodiums are the same way. They come in through this open channel, and they're like, oh, there are negative charges all over the place here. I'm not going to stand here where I came in. I'm going to start moving towards those negative charges. And this is what allows for the greater potential to be conducted, right? It gets conducted in both directions, as you can see, from where it was originated. Because as those charges came in, again, they were attracted to those negative charges on the inside of that membrane. All right, it's negatively charged on the inside of that membrane in both directions. Now, as, again, actually terminology, that also would be current, right? When we have the sodiums flowing, you know, down within the intracellular fluid here, kind of flowing away, that would also be an example of current. But what we also have to appreciate here now is that once those sodium ions left from this area here and came into the intracellular fluid, well, this area now on the outside face of this membrane becomes more negative all of a sudden than it was. And so again, these positive charges out here on the outside face of the membrane, they say, well, hey, now there's now this negative region here around that channel. We're attracted to that negative charge, so they, as they show with the arrows here, they shift over to make this area more positive. That leaves this area more negative. And so now these positive charges that are flowing down here say, oh, wait, there are negative charges further down within the intracellular fluid, but now I also perceive negative charges on the other side of the membrane too, so there's like pretty girls everywhere. Where do I go? Well, some will keep flowing down within the cytoplasm, and others will cross the membrane. They'll follow those negative charges that are there. And it's the ones that go in this direction, the ones that cross the membrane attracted to those negative charges, they are responsible for the decremental part. Because as soon as those positive charges flow back across the membrane to where they started, we're getting back to the resting potential now. We're putting the positive charge back on the outside, and it's relatively negative on the inside again. So that's the decremental conduction. So this is what it looks like if you could kind of, you know, measure it. Right here we have our membrane potential, right, plotted as a function of the distance from where we induced a depolarization. You can see that the maximum graded potential is right where that channel was, right where we induced that depolarization. But then notice how that positive membrane potential, that more positive potential, is sort of spreading out in both directions, but its magnitude decays as it goes. So it's spreading out because of this movement here. spreading out because those positive charges sense that there are negative charges throughout the cytosol here and they follow them, but it decays because as negative charges appear in the extracellular fluid, some of those ions will cross back through the membrane and they'll return to their original side of the membrane. And what you'll notice here then is that they kind of show us in this figure that if we get about 2 millimeters away from where the depolarization was induced, about 2 millimeters away from where we had that gated channel that we tripped open with some stimulus, by the time you get about 2 millimeters away, you don't even see that gradient potential anymore. We're back to the resting value. So I mentioned earlier in the definition of gradient potentials, we said gradient potentials are a local phenomenon. That's what we mean. Because they decay as they travel away from their site of origin, they are very much localized within the neuron. Let me, I guess, end with this here. This figure is showing us a neuron here, right? And so we can see that it's being stimulated right here. And we can see they've got these little recording electrodes that they've shoved in to various locations of the neuron. What you'll notice here is that when you stimulated this neuron right here, right, you'll notice that the recording electrode in that location close to the stimulus, it saw this big gradient potential. And note here they've marked on this figure the threshold potential, which is the potential that is needed to get us an action potential. And in this location of the neuron, very close to where the stimulus occurred, the gradient potential was above the threshold. Now, of course, the concept of threshold potential doesn't matter out there, right? It only matters in one spot. It matters here at the axon hillock, the axon initial segment, what they're calling here the trigger zone. And see what happens as this gradient potential is traveling from here where it originated to that axon hillock, axon initial segment region. You can see by the time it gets to the middle of the neuron, it has decayed, so it's just barely at threshold. And then by the time it actually gets to the axon hillock, the axon initial segment, it's actually decayed, so it's now below the threshold. And as it says here, you get no action potential. So this is one of the consequences of decremental conduction. We had a suprathreshold gradient potential way back here at the dendrite where we stimulated this neuron, but by the time it actually got to where it matters, the axon initial segment, it had decayed and became subthreshold, and no action potential occurred. We're going to talk when we come back Tuesday about, well, if decremental conduction is a thing like this, then how the hell do we ever get an action potential? If gradient potentials just decay and become subthreshold, how do we ever get action potentials? That is what we will talk about on Tuesday. So enjoy your holiday weekend. Try not to work too hard. Take a break from the short communication maybe for the 3 days of the weekend, you know. There's no quiz to worry about next week, so rest. Enjoy the holiday. Eat some turkey. Eat some for me so I don't have to eat as much. I will see you when we get back on Tuesday.